Okay, good afternoon, everybody. So I'm going to give you a little bit of the context of innovosexing techniques, some points about the current and future developments. So first of all, okay, I don't think it works.
Down?
This is.
Down one? Yeah.
Oh, okay. Okay, so I'm going to say a few words about the productions of table eggs, but also the production of foie gras, essentially in France, the methods that are used to kill one-day-old chicks, and some information about alternatives. So first of all, the issue comes when the phenotypic and physiological characteristics on one sex does not meet the productivity criteria or the product quality. So this is the case for the Gallus gallus species, essentially in the laying hen production that are produced to produce table eggs, and that were selected for decades on egg quality, but also on egg laying performances. So the breeders, parental breeders, will give, fertilized eggs, which contains, I don't know, I'm going to change my hand maybe. Okay, is it better? No. Okay. Okay. So the parental breeders will give either an egg containing a male chick, or an egg containing a female chick. So the female chick will give the further egg-laying layers, I would say, and while the male chicks will be culled one day after hatching, because they do not produce any eggs and their meat is of low yield and low quality. So this issue was concerning in France about 50 million chicks per year. We have the same problem related to the foie gras production, where the female have not added value. I mean, only the foie gras from male chicks are added value and are served as foie gras. And the female do not have good foie gras quality, and the yield is also bad. So it concerned about 15 million ducklings per year in France. So what we were doing before the decree and before legislation, the sexing of one-day-old birds was made using anatomical differences in the cloaca, but also differences in the size of the feather or feather colors. So here you have some pictures related to female feather and male feather, and then you have some differences related to the color of the feather. For the ducklings, there was some ostosexing strains that were developed based on an eye color, which is red for females and black for males. So the sexing was quite easy after hatching. And it has to be noticed that 50% of male ducklings are rare to be sold as farm counts for roasting, essentially for export markets. The methods that are used for culling chicks and the fate of dead birds, so they are based on the recommendation by American veterinary medical associations. So it's quite violent to imagine that these chicks were grinded or crushed using a specific dedicated equipment, but they can also be killed by gas inhalation using carbon dioxide. And after carbon dioxide treatment, they can be sold as for wildlife parks or animal rescue associations. But after grinding or crushing, they can be used also for pet food. So this awareness about the fate of chicks was highly publicized in the recent years, and it was followed by ministerial announcements to ban the killing of chicks in various European countries. It started with Switzerland in, sorry, I'm so bad with that, in January 2020, followed by a German announcement, and finally a French announcement that banned the culling of one-day male chicks last February 2022. Some other countries are quite interested also by the strategy. What about the foie gras industry? So they are not concerned by the decree, the French decree, which only concerns a Gallus gallus species, so chickens. And but already they are starting to think about stopping the culling female ducklings by 2025. For foie gras production, France accounts for 73% of the production. So 3 possible alternatives have been developed. Rearing brothers of laying hens, so using actual layer strains. But the problem is that there is no market today, but still there is some initiatives in progress. In Germany, they export male chicks to Poland to do some sausage and reconstituted meats. We can also develop some dual-purpose crossbreeds where the male could be reared for production of meat and female for production of egg. So it's in development for organic production, but it's essentially high cost. The quality of byproducts has to be optimized, and it's hardly applicable to conventional channels at present. What about innovosexing? So it has been deployed in all 5 hatcheries in France. The thing is that they wanted to select these specific alternatives because there is no major change in the organization of the egg industry, no need to develop new markets in the broiler sector, poultry sector, and the concentration of the efforts is made at hatchery level. So a little bit about innovosexing methods, some physiological points about what we call sexual dimorphism in ovo, some principles and challenges of ovosexing techniques, and an overview of ovosexing methods using the French case. So what about the principles? So the development of ovosexing techniques is based on the detection of differences between eggs containing a male embryo or a female embryo. That's what we call the sexual dimorphism. So we can look at genetic biomarkers because the sex chromosome is different between male and females. Some anatomical features, the development of the reproductive tract is different. Phenotypic traits, feather color, as I said before, but also we might have some indication about the growth kinetics that might be different also between male and female embryo. Some molecular characteristics, the lipids might be different, the lipids that are assimilated by the embryo might be different, the proteins, genes, hormones, but also metabolites and also volatile compounds. So the thing is that some of these features results from the activation of the genome and also the activation of embryonic metabolism. And some of them are visible, detectable lately during the incubation process. So the sexual dimorphism starts with the inheritance of parental sex chromosomes that are associated with some specific genes. So here you have, for example, the male is characterized by ZZ chromosome, while the female by ZW chromosome. So here you have a representation of the reproductive tract of the females. Here you have the yolky follicle that are developing at the ovary site. So this yolky follicle has the germinal disc corresponding to chromosome from the maternal origin. And then there will be some fertilization at this part of the oviduct by the spermatozoa containing the paternal Z chromosome. So from that, there will be the activation of the embryonic genome. Okay. So after the activation of the embryonic genome, you will have some different specific molecules that will be secreted throughout incubation. So it is believed that from the day of lay, there will be an expression and secretion of molecular biomarkers that might be different between males and females, followed by the egg pigmentation, but also the development of the reproductive tract and the appearance of a feather. So it's a little bit different for the Peking duck because the incubation time is about 28 days. It's also a little bit different from the species that are used for foie gras production, with the Barbary and Mula duck, which are related to 31 days of incubation for this one and 35 days of incubation for Mula duck. So the kinetics is a little bit different. Okay, So what are the requirements? First of all, you have to be, once you detected some characteristics that are different between eggs, then you have to be aware, you have to identify which embryo is in the egg. So it's very important to have methods that allowed us to identify the sex of the embryo using at least one of the sex dimorphic features that I've talked about. Especially if you want to target some specific time of incubation. As I said before, some markers are only appearing lately during the incubation process. So the first step is to search for sex indicator biomarkers that differ between a male embryo or egg containing a male embryo or a female embryo. So you look at the embryo itself or also egg structures. And the second step is to validate the markers on a large number of eggs from genetically different breeds with varying characteristics. So you look at, it depends on the weight of the egg, the shape, the shell color, but also the conditions you use to store and incubate the eggs. The challenge is that you need to detect these differences very early as possible, ideally before 6 days of incubation, because there is still a controversy related to when the embryo acquires the ability to feel pain during embryonic development. The method has to be non-invasive because you don't want to collect anything so that it could affect the viability and hatchability of the embryo. And it has to be fast, accurate, effective, applicable in the field on a variety of strains, but also with acceptable cost for both professionals and consumers. So here is an overview of the methods that have been developed worldwide. And in green, there's only 6 techniques that are actually marketed. So I'm going to talk about all these techniques. So first of all, the plant egg techniques is based on sexual dimorphism that is associated with sex chromosomes. So here, usually they used the technique at day 9 of incubation. And the idea is to detect the genes located on Z or W sex chromosome. So for that, they need to have cells containing DNA, excuse me. And the characteristics is that they collect cells that are contained in what we call the allantoic fluid. So it's pretty invasive, but the advantages is that you can do that with all breeds, actually. The other constraint is that the allantoic fluid does not develop until the 6 days of incubation. And there are other structures that contain DNA before day 6, like the embryo or the yolk sac. But you can imagine that it's pretty invasive if you want to collect the cells from here. And it should affect viability and hatchability of the embryo. Auburn developed a technique based on magnetic resonance imaging, MRI. The characteristics of these techniques is that it's non-invasive and you could apply that to all breeds. The idea is to detect some differences in the development of the reproductive tract. So here is an example of the male reproductive tract with the 2 testes and the differences that you can see at the ovary side for female. So the thing is that these differences, you can detect them after dissection, I mean visually as early as 9.5 days of incubation, but actually the techniques is now quite efficient after 13 days of incubation. Okay, what about AAT and Nectra? They've developed similar techniques to detect the color feather. So here you have the female and then you have the male. This technique has been developed from day 13 of incubation onwards. It is based on hyperspectral methods. It's non-invasive, but you can apply that only to brown breeds that are sexable using the feather color. You have another techniques based on the molecular differences, looking at genes, proteins, metabolites, lipids, and etc. So you can maybe see some differences in yolk and egg white assimilation that might be different between male and female embryo because they might have some different metabolism. The thing is that the embryo, when it started to develop, will secrete some specific molecules that can diffuse through the yolk, but also through the eggshell pores, and that you can detect on the surface of the eggshell, for example. All these molecules can be also secreted in other compartments of the egg, including some extraembryonic structures like the amniotic sac, which is here, the allantoic sac, which is here, and which contain the allantoic fluid. So this is the strategy that used Select to develop his techniques. So the idea is to quantify a specific hormone that is only secreted by female, which is estrogen sulfate, and they do collect some allantoic fluid. This technique is it's semi-invasive because you have to collect something from the egg, but the advantage is that it can be applied to all breeds in theory. And the thing also is that the allantoic fluid does not develop until the 6th days of incubation. So what about the French case? Because this is the one that I know the most. So as I said before, the decree was published last year on the 5th of February. It prohibits the killing of chicks of Gallus breeds that are rare for table egg production. But there are some breeds and some specific exceptions that are out of the scope of the decree application. For example, the other bird species are not concerned, for the ducks, for example. The chicks that are used by the pharmaceutical industry, for example, the vaccines that are produced against human influenza requires the use of fertilized eggs to allow for the proliferation of the virus that will serve for vaccine production, but also those that are used for veterinary diagnostics. And the chicks that are hatched for animal feed purpose, the chicks that are injured or having a disease likely to cause suffering to the chick, hatch male chicks that result from sexing errors, because there are some sexing errors with oversexing techniques, and chicks from right breeds for which in-oversexing was lacking at the time of the decree publication. So the decree also stipulates that the equipment that has been chosen by hatcheries cannot be questionable for a period of 5 years. And the methods that have been selected by hatcheries need to be applicable for fertilized eggs prior to 13 days of incubation. So I'm sorry about this table, but it's only to show you the comparisons about all the techniques that have been chosen by all French hatcheries here for chickens, but also for duck. So you can see here that most techniques can be applicable from day 13 of incubation onwards, except for the duck species, where the appearance of differences in the eye color is much more earlier. You can see also that some of these techniques cannot be applied to all breeds, especially this one, for example. Only the breeds that were sexable on the feather color. This one used by Nextra is also sexable on feather color. The speed might be quite different, especially for this one, 3,000 eggs per machine per hour, in comparison with the two other ones, which are much more efficient in that kind of activity. And hatchability can be also impacted by the techniques. The objective of all these companies are to improve the precision, but also the speed, and to improve early detection. So here are the different hatcheries that have chosen these techniques. The fate of sorted eggs, once you have determined which egg is containing a male embryo, you have to sort it from the incubator. So we call that hatchery byproducts. So hatchery byproducts encompass stillborn and killed chicks, but also eggshells from hatched chicks, unhatched incubated eggs, including eggs that are sorted after in-ovosexing, clear, what we call unfertilized eggs, but also those that have been stored but not incubated. So it represents several tons of solid waste, the recovery of which is subject to governmental and health regulation. So according to European regulation, clear eggs, incubated eggs, and certain other byproducts, including the chicks, the eggs that have been sorted, are classified in category 2 because they are low risk, they represent low risk to public health, they can be used as organic fertilizers for composting or conversion into biogas. But the direct use of these bioproducts on soil and in compost or biogas production without prior pressure sterilization is strictly forbidden. So some hatched chicks may fall into category 3, particularly if they have been slaughtered because they could not be used in the human food chain, so male chicks of layered strains, for example. They may be used for animal feed after carbon dioxide inhalation and freezing. That can be sold to protected to feed protected animal species, animals that are hosted in zoo, parks, or wildlife protection associations, or even for certain privately owned animals. So it concerns essentially reptiles and birds of prey. So this derogation related to the French decree is included in the decree of November the 4th last year, and concern in particular any chicks whose embryo cannot be sexed by the color of its feather. So the question of embryo suffering and the necessity to develop early in vivo sexing methods. The question is related to the onset of the ability to feel pain that remain controversial. For example, if you look at the appearance of the first heartbeats, the motility, touching, spinal cord connection, taste, hearing, essentially also the oscillographic activity. So it's quite controversial. And the controversy depends on the parameter and indicators that are used to identify pain in the embryo. So the consensus, I would say, feeling is that it seems that there is an absence of consciousness until day 15 of incubation, and there is still a gray zone between 8 and 13 days of incubation. So 15 days of incubation, it's quite late during the incubation process. So it remains essential to have some more knowledge about the appearance of pain during embryonic development to increase the acceptability of oversexing methods. The embryo that are more than 80% developed, corresponding to more than 16 days for chickens embryo, are considered as live animals. There are some approved euthanasia techniques, such as cooling at 4 degrees or 20 degrees for 4 hours, freezing embryos, or anesthetic overdose or decapitation for this kind of embryo, which are quite violent pictures to imagine, actually. What about the other developments? So the thing is that, as I showed you on the table, most techniques are applicable from day 13 of incubation. If you look at the timeline of development, you can see that even from day 8 to the end of the incubation process, the embryo looks like a chick. So the earlier, the better. If we can develop some specific early oversexing method, it would be better for everybody. So this is what we are working on the PPILOW project. So this is my part. Actually, I'm more related to the physiology of development. So my task is to identify some early biomarker of sex in vivo, focusing on embryo that are developed before 8 days of incubation. This process is invasive at the first step. I have to collect all the different structures of the egg to identify the sex of the embryo and then try to correlate the result I have with the sex of the embryo. So the objectives are to provide a list of sexual dimorphic molecules, genes, proteins, lipids, and etc., and to identify which egg structure is the most different between male and female containing eggs, and which structure is the more relevant to focus on to develop such early in vivo sexing. The second part of this work package is related to the development of a non-invasive oversexing techniques using radio frequency. So here are the wavelengths of the radio frequency, and the advantage is that it's non-ionizing, so it's not harmful, and also it is expected to be of low cost. There are some other techniques like sex reversal skewed in favor of females. So this company is looking at different, I would say, incubation parameters, looking at the temperature. It's a combination of temperature, humidity, carbon dioxide level, but also during storage, and sound vibration. And they think that they developed this system that affect the sex development process in poultry embryo and turn genetic males into functional females chicks. So they say that during incubation, they influence the expression of genes that are responsible for the development of the reproductive system, and they turn male embryos into female embryos. The other technique is gene editing. So it's using GMO-derived products. So the advantage of this technique is that you can sex the embryo before incubation, so that there is no development of the embryo. Using specific light, you can easily detect the egg containing the male embryo. There is some concern about the acceptability of a GMO-derived product. And the thing is that the production will depend specifically on edited strains. So what about the take-home messages? So in vivo sexing methods are based on the Z and W sex chromosome differences that can be looked as before incubation, based on anatomical differences, the gonad development of reproductive tract development, phenotypic differences, molecular differences, but also we are looking at metabolic differences between males and females. The thing is that male and female differences appear at specific stage of development, which may limit the possibilities of using certain oversexing approaches at earlier stages, since differences are not visible earlier. There are several in vivo sexing studies in development and improvement that are in progress, and the two other alternatives are still investigated, like rearing brothers of laying hens and the development of dual-purpose crossbreeds. So this review actually will be published in December in a French journal, and the English version should be available during 2024. Thank you very much.
Thank you, Sophie, for this overview about the in vivo sexing methods. And in the PPILOW project, in fact, there is one specific activity dedicated to the stopping the elimination of the layer male chicks. And one part is about the development of such a technique, and the other one will be dealt with the next speakers about the dual-purpose breeds. So now the discussion is open for about this presentation from Sophie, if you wish.
Thanks for the nice overview. I wonder if you have any information on the cost of the methods that are in existence?
No, actually, this is not exactly my part, but I think I can give you some names of a guy who is working at ITAVI, which is a French institute. He's very specialized in this kind of question, actually, but I have no idea. Sorry.
Yes, perhaps we can add that within the project there is based on this activity, there will be also the construction of a business model by the colleagues of Tunen Institute dealing with this kind of methodologies. So probably soon we will be able to provide also some data about this. Are there other questions?
Thank you. My question is, what is the fate of the eggs sexed as males?
Yeah, actually you can use it for compost or for other things, but it's quite difficult because you have a mix of different things. You have eggshells, you have remaining yolk and white and embryo and extra embryonic membrane. So depending on the countries, I think you have specific regulations how to process this material to use them as fertilizers and etc.
Thank you. Thank you. Other questions?
From vertical point of view, your comment is better the first in ovosexing or continue with the dual purpose? Or what is your...
I'm not sure I can answer to this question because I have a personal point of view, but I am here for, in the name of INRAE Institute, and I don't think I can, maybe in personal we can talk a little bit about it.
What we can tell about this is also that definitely Sophie and colleagues are trying to go as early as possible to be really on the very first steps of incubation. So this is one point related to the ethics and the pain, because obviously some of the methods now are quite late, well, in the middle, after the middle of incubation. But yes, this is really an ethical point we have to tackle, taking into account also the social expectations, the economic point of view, environmental, and yes, and obviously, yes, for some, perhaps we can go further in the discussion after the presentation of